Assalamualaikum everyone, I'm Afiq and today we're going to talk about digestion. Digestion is the process that breaks down large and complex food pieces into smaller and simple pieces that can be dissolved for an easy absorption. There is two types of digestion, which is physical digestion and chemical digestion. The main difference between these two types of digestion is that physical digestion involves mechanical digestion that breaks down food into smaller pieces, while chemical digestion is a decomposition process of food complex molecule into simple molecules. There is four organs that involve in digestion: mouth, stomach, small intestine, and large intestine. Let's look at mouth. In mouse, food undergone two types of digestion, which is physical digestion and chemical digestion. Under physical digestion, food will be chewed until it forms a lump called bolus, and under chemical digestion, salivary amylase will hydrolyze this starch into maltose. The suitable pH level for salivary amylase to work at optimum level is between 6.5 and 7.5 pH. In other words, at neutral state. And then food will be transported into stomach through esophagus by peristalsis. In stomach, food will be digested by gastric juice, which is secreted by gastric gland, which contain hydrochloric acid, pepsin, and renin. Hydrochloric acid will provide acidic environment for pepsin and renin to work at optimum level. Stop the enzymatic action of salivary amylase and kill the bacteria. Pepsin will digest protein to polypeptide, while renin will turn soluble milk, which is carcinogen, into insoluble milk, casein, and then the casein will be digested by pepsin. After that, food will be churned by the peristaltic action of stomach wall muscle for a few hours. The content of stomach will be turned into semi-fluid that's called chyme. Chyme then will be transported into duodenum. In duodenum, there is two main sources of enzyme, which is the first, pancreas. Pancreas secretes pancreatic amylase, trypsin, and lipase into duodenum through the pancreatic duct. And then we have liver, which produce bile. The gallbladder will store bile, and then the bile will flow into duodenum through the bile duct. The function of bile is neutralize acidic chyme, prepare an alkali condition, which is between 7.6 to 8 pH for enzyme action in the duodenum, and then emulsify lipids by breaking down lipid into tiny droplets to increase surface area for lipase activity. After that, pancreatic amylase hydrolyzes starch into maltose, and then trypsin will hydrolyze polypeptide into peptides, and lipase will hydrolyze lipid into fatty acid and glycerol. Glands on the ileum wall secrete mucus and intestinal juice that contains maltase, sucrase, lactase, lipase, and erepsin. The alkali medium in the ileum allow enzymes to act in its optimum level. In ileum, maltase hydrolyzes maltose to glucose, and the sucrase will hydrolyze sucrose into glucose and fructose, and lactase hydrolyzes lactose into glucose and galactose. For lipid digestion, we have lipase that will hydrolyze lipids into fatty acid and glycerol, and lastly, protein digestion, which erepsin hydrolyzes peptides into amino acid. So basically, that's it. What happened in our digestive system. And lastly, the food will be transported into large intestine, which then in large intestine, the dead cell, undigested food, and fiber will be turned into faces, while water in the large intestine will be absorbed. So that's it, and that is the end of the video. Thank you so much for watching. Sure you subscribe and like this video. See you again very soon. Bye bye.